So this plan my talk, which is a little bit different from what I have promised in the title, because I want to talk on some scientific issues in more detail. So the first part of my talk about the computational labs in Vietnam. Then I move to the main factors that govern fiber information process, and then about drug design for Alzheimer's disease, and the last part is about future work and some possible collaboration. To my best knowledge, we have 4 labs working on computational biophysics and medicine. The oldest one was established in 2003 in Hanoi, and the director of this lab is Professor Suan, and this lab mainly works on protein and membrane folding using some kind of post-grain models. Another lab by Thai Minh at the University of Medicine and Pharmacy. They work on silico and vitro drug design. They have some targets like anti-tumor, or they claim they also work on Alzheimer's disease. I'm not sure. And the lab of Professor Wong Sung at VNU, and I think that now they are focusing on membrane proteins, also using some kind of post-grain model. And I should talk in more details about our lab, not at VNU, but at the Institute for Computational Science and Technology, also in Ho Chi Minh City. Let's look at these numbers. The number of members of our labs between 5 and 15, and so our lab is the largest one, but not necessarily the strongest one. And about this publication for last 4 years, this is not so many publications. But if we divide, for example, we take this last year and divide this number of papers by number of professors, 4 professors from 4 labs, and you get something like 3 papers professor per year, and by economic standards, I think that's not so bad. It would take around 2,000 papers by the whole economics scientific community by last year, and divided by about 5,000 professors in Vietnam. And it is something like 0.4, and I think that is reflected in this overall publication for certain Asian countries. Here you see that Vietnam is here still low compared to Thailand, Malaysia, or especially to Singapore. So I see that because of this still poor situation, we are getting today here to discuss somehow how to promote one piece of science in Vietnam. Okay. Now a little bit about our computational Institute for Computational Science and Technology. This institute is run by overseas Vietnamese scientists. I mean that all lab leaders are Vietnamese who work outside, like me in Poland, or some other from the US or other countries. And we have 5 labs, and we mainly work with students by Skype. Every year we come here for 1-2 months to work together with students. I want to express my thanks to VNU, who strongly support our labs, that all of my students or collaborators have graduated from VNU. Now, about activity of our lab which of two groups, my group and Dr. Lille group. I'm not sure that he came today. So we work on protein folding problem, on stretching or pulling proteins by mechanical force, and concerning bioinformatics, we do homology modeling, sequence analysis, and we work on this. Our most favorite subject is influenza A virus, and but today, to make the close connection with the activity of Professor Eisenberg, I will talk more on this protein integration and drug design for Alzheimer's disease. Maybe it's not a good idea for audience because we have some strong overlap between two talks, but maybe good for understanding each other, especially with Professor Eisenberg. So you know that normally protein form into the native state. If so, you don't have any problem because protein should have normal functions. But under some situations, protein can get together and form some kind of aggregate or oligomers. In native state, maybe you have very organized structure like fibers. So one of this structure is amyloid, and Professor Eisenberg have talked a lot about this. So again, this I repeat that the bad thing is that protein integration is associated with many human diseases. One of them we today my talk is Alzheimer's disease, which related with, as you know from the previous talk, with A-beta peptides. Now, I think that I should skip that slide. Here, brain of a healthy person, and here, brain of a person with advanced Alzheimer's disease. And you see that you have some damage here because the formation of amyloid plaques. Yeah? This, I think that is still hypothesis because nobody knows what is the cause of this disease. We just assume that Alzheimer's disease is related just to formation of fiber formation of A-beta peptides, but this is not confirmed. Now, about these structures, you see that here you have different sequences, but they share very common structure, that all of them have cross-beta structure. Yeah? So then the question is, maybe there are some universal factors that control this fibrin formation process. I have to say that in the previous talk, Professor Eisenberg focused on structures, but in my talk I focus on kinetics. Now, there are several factors like hydrophobicity or beta propensity. Let's look at this. Look at these beautiful experiments. They have just 4 amino acid peptides. Okay, and here you have 2 phenylalanine. And for this short peptide, you have a nicely picture for fibers. Okay, but if you mutate these 2 phenylalanine by alanine, this means that you reduce hydrophobicity of your system, that you have empty picture here. This means that no fibers is formed. So hydrophobicity plays a very important role in fibrin formation. And it's clear because if you have peptides highly hydrophobic, then you know that they scared of water. So they try to come together inside water, forming of aggregate. Yeah? Now another factor is charge. If you have a lot of charge, then due to electrostatic repulsion, they cannot come close to they cannot come close, and then you don't have probably you don't have fibrin. For example, for protein S6, here we have 4 charged residues, and in this case they don't form any fibers. But if you replace these 4 charges by 4 hydrophobic amino acids, okay, amino acids, then people have seen albumus, and this means that this charge plays some role of gatekeepers, that prevent this protein from aggregation. Today I want to convince you that one of the most important factors is that the population of fibrin prostate and star are just in monomer state. Because our group worked a lot on this problem. Definition of fibrin structure and star is very simple. You just take this fiber structure and structure of monomer in this fiber state is structure of N star. Yeah? Just monomer structure in fibril state. And now, if you have barriers between native state and N star or excited state, if this barrier low, this means that population of this N star stay high. And in this case, you have more chance to have fibers, because you can imagine that if barriers is high, then it's very, very difficult to activate your protein, come out from the native state to form some fibers or to another state. And it was confirmed by simulation and by experiments. For example, A beta 40 has very little beta structure compared to A beta 42. But if you have high high content of beta, then this means that the population of N star high. And in this case, you see that as shown from experiments, A beta 42 form fibrin much faster, or 1,000 times faster than A beta 40. So in this picture, you can see that the main experimental result about the factors that control fibrin formation. So by mutation experiments, Dodson group from Cambridge, they have shown that fibrin formation rates here are strongly correlated with the hydrophobicity, charge, and population of N star. And it took us many years to explain this experimental data. Yeah? What is our problem? It seems that very simple. On computers, you just start from some random configuration, and you run long enough to get into fibrin. Yeah? But it's not so, because you see that the fibrin formation times is upward of days or maybe months. But on computers, you can run something like a microsecond. Especially in Vietnam, I'm not sure that you can do it easily. So one has to. So we have a huge gap between computational facilities and real life. And so we come up with a very simple lattice model, which allows us to speed up calculation. This our model is very, very simple. This each peptide has only 8 amino acids, and this is a simple force field. And by this simple model, we can capture beta structure like this. This is a beautiful picture, okay? Or fibers. And now, using this simple model, you can see the difference between folding time and fibrin formation time. You see that folding time is very fast compared to fibrin formation. So even by this simple model, you can capture the fact that folding time takes around microsecond to second, but fibrin formation time days, as I said, per month. So So somehow you just qualitatively capture the picture of this process. Now we want to look at the population of N star. So because of this shortcut time, you can do exact enumeration. So here you have 65 sequences, and you can do exact enumeration. You calculate this population of N star state. And these are final results for dependence of fiber formation in our model on charge, hydrophobicity, and population of N star. And you see that you can nicely reproduce this experimental data by Dobson group, and here for physicists probably very nice exponential dependence of fiber information type on this population of the N star. Now I want to make Professor Eisenberg happy by this slide. Namely, he is the first to introduce the concept amyloid. So if you have many students, you have genome, okay? But if you have many amyloid peptides or proteins, and you have some kind of amyloid, this means that amyloid is a universe of proteins that are able to form fibers. That's his concept. And his main message from this paper is that not all the protein or peptides can form fibers. This is a very interesting question, and we were very excited about this and try to understand this question just using our simple model, very simple model. What we can do it? As I said, we use this 8 amino acid model. Okay, so you can in total we have only 65 sequences, but from this you have only 6,000. Sequences that have unique ground state, and you keep only these 6,000. Then you calculate population of N star for these 6,000, and you see that only 200 sequences has population bigger than 1%. Then we assume that only those which have population more than 1,000 can form fibril within reasonable time scale, and this somehow explain or somehow support the hypothesis of Professor Eisenberg. So I think that this for us is a small piece of little bit beautiful work on this subject. So recently we have implemented some new method to calculate the size of critical nucleus, but I don't have time to talk about this. Let me jump to another topic about drug design problem for Alzheimer. So because most of you are students, so let me remind you what is the drug design problem. So for us, it's very simple. You have interaction between drug or possible drug and drug target or receptor. Okay, and we have to calculate the binding affinity of ligand to this receptor. And before to do this, let me show you this drug discovery process, which involves several steps here. So this process long, pretty long, 10 or 15 years to design one drug, and it costs around $1 billion US. So how to shorten this process and make it cheaper? And one of way to do this is to use computer. So what computer can do is just to speed up this lead and optimize identification and optimization. And even by this simple approach, people have discovered, I think that 5 ligands before they have been approved by FDA as drugs. So still this approach still useful, and we always follow it. So about binding affinity, here you see that you should calculate the binding affinity for complex receptor ligand, and you subtract this binding affinity of receptor ligand, and if this quantity is negative, then you have a good chance to have good candidate. So today you have around 40,000 of all possible drugs, and they are more or less small, and their binding affinity around -9 to -12 kilocalorie, and with inhibition constant between micro and nanomole. And I think that many groups around the world working on inhibitors for A-beta aggregation. So typically around 400 compounds are under investigation. So many of them, many kinds of them here listed here, but our group just focus on short peptides and natural products. Yeah? So this to show this short peptide, we take very short peptides, just 3 amino acid peptides. So for this you have all possible peptides, 8,000. Okay? When you look into this target, these fibers and monomers, but people always ask me, do you know where is this binding site? I don't know. But on computers I know, but in real life I don't know. And no body has confirmed where is the binding site. Okay? But for this repeptide we have shown that if you have many rings like proline or tryptophan, then you have very good binding affinity. And our simulation has been confirmed by experimental data from Slovakia group, and the best from this 3 peptide have inhibition constant in the range of micromole. This means that it's good for drugs, but there's a long way to go if you can use this. Now, another subject is about natural products, and for simulation, we took compounds from Taiwanese database. This address is here. Actually, they have around 36,000 compounds. Then our strategy is to use some Lipinski rule to reduce its numbers. Okay, to reduce its numbers, and then we do docking for this smaller set of compounds. And after docking is not so good, then we use more precise approach like MPVSA in molecular dynamics simulation, and we get around 5 very good compounds. But our collaborators from Taiwan, they could purchase only 1 compound. This by name dihydrochancol. This has 2 rings. It's very similar to curcumin, which is undergoing the second phase of clinical trials. And you see that without this ligand, you have fibril here, fibers. Okay? But after adding this dihydrochancol, you have clearance of fibers. Okay? So, and they have measures of the toxicity. It turns out that fortunately this compound doesn't kill cells. So in this prospect is very promising. And I would say that we have calculated the this blood-brain barrier and this can cross, because to deal with Alzheimer's disease, your ligand should go to brain, okay? So it should be able to cross some barrier, and this compound can cross barrier easily. So, but from our calculation, again. Now about future work and possible collaboration, we are very excited about in vivo activity of this compound, okay? And our collaborators are applying for some grant to get money to do experiments with C. elegans. And in my group, we also work on the interaction between kappa and A-beta peptides. This is important. From experiments, you know that the kappa can speed up aggregation and make the system very toxic, but nobody can prove it by simulation. So we are trying to some classical force field to speed up calculation and to see if really kappa has high impact on the fiber information process. And another project on the mutation effect on aggregation like this, we are working in close collaboration with the French group in Paris, and we look at the effect of environment like pH, how it affects the morphology of fibers, and so on. So this another project about breast cancer. I will say that I have no idea about breast cancer, but recently Dr. Suan and Prof told me that for this breast cancer, they have very good target, like so-called steered molecular dynamics. What is this? So you see that we apply external force to pull this compound out from the binding site, okay? And during this process, our ligand experience some force, and profile of the force look like this. And if this force is high, then we think that if you need more force to rupture this system, this means that you have good binding. So based on this hypothesis, we predict that around 10 compounds here are better than this reference compound which have inhibition constant of 25 micromole. So this is some typical top hits for CD44, and we hope very much that Dr. Fook and lab can find some people or money to work on this hit. This picture about our overseas collaborators on Alzheimer's disease, Philippe Dremot from Paris. We do simulation together. But these three groups, they work on experiments. They check this box, they measure emission constant. But maybe after this conference, I hope very much that we can initiate some collaboration with UCLA, and maybe more collaboration with more groups in Vietnam, I hope. And on this slide you see my collaborators. Many of them are very young and ambitious, and thank you for your